So good afternoon, everyone. Today, and welcome to the Young Investigator seminar series. Today, we have Oleg Lunov. Oleg, he's a graduate. He graduated in Ulm University in Germany, and he did his postdoc in the Pharmacological Institute there at Ulm as well. And then he moved to the FCU in Czech Republic in Prague, where he's been there already for a long time, right, Oleg? You've been like how long in total?
From 14 years.
Yes. So 8 years. So you are already very, very senior there. So it's a very nice institute. They have very nice technology, microscopes, and he's gonna show us very nice images for sure. So if you want to proceed, Oleg, thank you for again giving us the opportunity to get an update on your research, and you can start whenever you want.
Thank you very much for kind invitation, and I will start sharing. And I would like to try to entertain you a little bit about the nanoparticles, the thing that is quite often omitted in terms of liver interactions, and but quite interesting. And there are some drugs available. A little introduction, as Javier told, I'm located in Czech Republic in Prague, in Institute of Physics. Basically, you may think what has to do Institute of Physics with biology, but we have a broad range of research interests in our institute, starting from the cosmology, ending up with the cell biology, which is done in our lab of biophysics. We devote our research to studies of different physical cues, how they affect in the living cells. We study non-thermal plasma, laser irradiation, how the mechanics of the microenvironment affects the living cells. Thanks to Cost Action, we got involved in very nicely in the consortium was working on organoids. Our technology, I hope, helps to understand the organoid development more deeply. We study a lot of nanoparticles, how the nanoparticles affecting, mostly dealing with the cells from the liver. We try to integrate an approach of taking from one hand the physical methods and physical characteristics, merging them with the biological techniques and putting in the center the cellular models. In this way, we are aiming to get the platform for evidence-based physical treatments, because there are a lot of physical-based treatments like laser therapy, magnetic therapy, electromagnetic therapy, etc., that are trying to invade the clinics. However, they do not have the evidence-based platform. And this is our far-going idea, and maybe we will reach it. And we are focusing on the molecular mechanisms, trying to merge really physics together and really create an interdisciplinarity and understanding between physicists and biologists, and introducing the really understanding of the molecular mechanisms. Because as evident for you that the even if the treatment or drug is working, we need to know and understand the mechanisms, because it helps in many things like listed. And we see that uncoupling those molecular mechanisms of physical cues, we will boost up the research in this direction. However, for the physicists, it's not always evident. As here, I presented some nice tips from the reviewer when we criticized one type of physical treatment, and I'm happy that we selected open review, and this is now available online, that the reviewer said that it doesn't matter how the things are working and present it as a kind of some allegory of baking a cake, that it doesn't matter how you bake it, it matters that it's finally done. We answered to that using actually this quote, that even the baking the cake, it matters how do you do that. The final quality is very much different. So molecular mechanisms matter. And now coming up to nano. Why is so interesting, and why so, I would say, overhyped sometimes this direction is? First of all, I will give some definitions and historical definitions. Actually, 10 years ago, nano was defined that there is certain size threshold at which materials start to behave uniquely, physically and chemically, than isolated atoms or molecules versus bulk material. And this threshold was defined as the below 100 nanometers. Nowadays, it's a little bit vague slide, but I would like to emphasize that nowadays the things changed. And first of all, no one is talking solely about unique or novel properties. It might be improved physical chemical properties of the material, and this threshold overall, the 100 nanometers, stayed, but depending on the authority, the borders of this nano scale dramatically differ. For EUPAC, it's 500 nanometers. For FDA, it's 1000 nanometers. EMA still sticks to the 100 nanometers. Another addition of the swirl disturbance in the definitions is the nanomedicines. FDA defines nanomedicines as basically nanodrug. But there are also definitions of nanomedicine as the discipline. There is also some kind of the not pure understanding of the definitions. The tempting idea that some materials of a size of single proteins or aggregates of proteins, but smaller, way smaller than the isolated cells, would impact biological system is eye-catchy, and various applications in biology emerged. Starting from research, we have labeling quantum dots, the gene delivery nanoparticles, then invaded the real market in the antimicrobial agents, in cosmetics, and quite a number entered the clinical trials and really resulted into nanoparticle-based medicines, nanomedicines. What are current state? Currently, we have 29 FDA-approved nanomedicines. Interestingly, in 2016, it was 51. Majority devotes to anti-cancer treatment. Then there is a inflammation, immune, and pain relief agents, then those infection and incremental other pathologies. The market is growing extremely, and it's interesting to look retrospectively that the prediction of the growth of the market are quite in line what we currently have. There are three main classes of the nanomedicines. Those are polymeric, classified based on the material which they made of. There's a polymeric, inorganic, and lipid-based. We try to understand how those different types of particles are interacting with the cells, how the cells are engulfing, digesting, and maybe there is an impact on intracellular pathways. We have a set of different materials, like polystyrene nanoparticles, silica nanoparticles as the model particles for the drug delivery or nanoparticle cell interaction, iron oxide carboxy dextrin particles for the as a contrast agents for MRI enhancement, And now we also are focused on very interesting novel nanomaterial DNA nanostructures that are getting a huge attraction from the biomedical perspective. Why iron oxide nanoparticles? I mostly will speak about those that are quite interesting, and there are a lot of interesting things happening with them. They are magnetic. They create one can create a stable water-based colloids and of them, and even they behave like a full fluid called ferrofluid. They found biomedical applications actively researched as antibacterial agents, drug delivery, gene delivery, cancer treatment. There is several drugs that approved for iron deficiency anemia, and what we mostly in our lab focused on the particles that are used as a MRI contrast agents for specifically even liver MRI imaging. Those particles are mostly made of iron oxides, like magnetite, amide, to a less extent hematite. There are several various coatings could be applied, and even from the very beginning there was the there was quite a lack of studies identifying the iron oxide nanoparticles as potential liver injury compounds. And quite a number of the compounds that reached the market were approved as contrast agents for MRI imaging. In fact, this is not only related to iron oxide nanoparticles, but in general, particles, when they enter systemic circulation, they are predominantly sequestered by the liver. There is a so-called kidney filtration threshold. If we create particles lower than 6 to 8 nanometers, then there is a tendency that they would be excreted by the kidney. However, even those, not all of them, the small, even those small particles are sequestered by liver. If we make up a sketch of the kind of average particle of iron oxide that is used as an MRI contrast agent, but we find that this is typically magnetite core of a size up to 40 nanometers, and with the carboxylic strain of some sugar shell up to 200 nanometers. So definitely sequestered by the liver, and the research showed that not only simply by liver, but the Kupffer cells in the liver predominantly, and even may retain for months over there. The logical question arises: how do macrophages handle those iron oxide nanoparticles? Firstly, we selected two types of particles that are having the same core size of magnetite of 5 nanometers, but different sugar shell, up to 61, and another one for 20 nanometers. Both particles were taken up by the macrophages, the concentration dependently and time dependently rather rapidly. Bigger particles, macrophages took to a higher extent than the smaller ones, and retained to the lysosomal-like compartments as revealed by the TM imaging. Using a series of the pharmacological inhibitors of specific pathways of the endocytosis, we revealed that both particles are predominantly taken up through the scavenger receptor and clathrin-mediated endocytosis. To a less extent, the macropinocytosis was involved. To confirm that the scavenger receptor and clathrin-mediated endocytosis is involved, we did immunostaining, showing that indeed both particles retained in clathrin-coated vesicles upon the engulfment by the macrophages. Then we downregulated the scavenger receptor and showed that the cells have, like in the scavenger receptor, they have an impaired uptake of both types of particles. Knowing the endocytosis mechanism could be a good opportunity to improve it. And as the particles are magnetic, then we use static magnetic field and could effectively increase the uptake of the particles by cells that are taking them to a very low extent. However, that process takes some hours. You put the magnet under the plate and wait. So we designed a certain coil that produces the huge pulses of the magnetic field, and using those pulses within a minute, we could increase the efficacy of the uptake of the iron oxide particles. Then we thought that probably we can use it more elegantly and load cancer cells with the particles. The particles goes to the lysosomes, and then applying this pulsed magnetic field induce disturbance in the lysosomal structure and induce lysosomal membrane permeabilization. Indeed, we could load very nicely lysosomes. Lysosomes got bigger, and then application of external pulsed magnetic field resulted into the damage of the lysosomes, release of calypsin B, and execution of the apoptosis. We could expand that even to the multicellular aggregates of 3 cell lines and see that the markers of the apoptosis are upregulated. However, we saw a limitation of that technology because the magnetic field is decaying very rapidly with the distance, and in order to create a sufficient force that is rupturing the lysosomal membrane, one need to achieve high magnetic field and the gradient of it. And it's impossible even with these multicellular aggregates to get that field in the center of it. So quite very fastly decaying with the distance. But let's go back to the macrophages. What happens as a functional consequence upon the uptake? In fact, after a short period of the incubation, there is nothing. However, after prolonged incubation, the macrophages start dying, and the smaller particles are more toxic than the bigger ones. Interestingly, that not all cell types are responsive. Endothelial cells, for instance, they were not facing cytotoxicity. The macrophages showed the markers of the apoptosis in terms of annexin 5 propidium iodide staining, and we confirmed that there is indeed an apoptosis by the caspase 3 activation assay. We hypothesized that the reason for the toxicity is the iron oxide core, and that is playing a role in the phantom chemistry producing the reactive oxygen species, and that is triggering the apoptosis further. So we confirmed that the particles, both particles, induce the excessive reactive oxygen species accumulations in the macrophages. The smaller particles show way higher levels of ROS. These ROS leads to JNK activation time dependently and concentration dependently, and faster it happens with the smaller particles. To confirm the involvement of the JNK, we used the inhibitor and showed that could rescue cells from the death using appropriate concentration of the JNK inhibitor. So basically, our hypothesis is that the iron oxide produces the ROS5 phantom chemistry that leads to the JNK activation, and that is executing the apoptosis further. Why the smaller are more toxic than the bigger? particles. The matter of the kinetics of the cleaving of the carboxylic strain shell. The macrophages take up the particles and retain them, do not excrete, as evident by the over time by the ion content. But the shell is decaying, and the smaller ones are unpeeled faster by the macrophages. There are plenty of the enzymes. One of them is alpha glucosidase that is located in the lysosomes, so then quite heavily presented in the macrophages. And the particles are colocalized with lysosome and colocalized with the alpha glucosidase. And interestingly, if we compare, let's say, macrophages with the endothelial cells, the level of the alpha glucosidase is higher in the macrophages in comparison to endothelial, which explains why the endothelial cells were not subjected to the toxicity versus macrophages were. And these also explain why smaller particles are toxic more than bigger ones. They have the same core but different thickness of the shell, and it's simply a kinetic process when the degradation occurs faster with the smaller particles. Whether that thing happens in vivo. So the particles are intravenously injected accumulates very rapidly in the liver of mice and retains for a long period. We tracked up to 21 days. They stay there and show the signs of the apoptosis in Kupffer cells, elevated caspase 3, active 1, the markers of the annexin 5 propidium iodide, and finally depletion of the macrophages in the liver cross-section. To boost up the role of the ROS and to show the that we can mitigate that toxicity process, we use the edaravone, which is a ROS scavenger, and we're able to rescue macrophages that are undergoing apoptosis with the ROS scavenger. So that brings us to our model of the mechanistic understanding of iron oxide particles cytotoxicity in macrophages. The particles are taken predominantly via scavenger receptor and clathrin-mediated endocytosis retain in the lysosomes, where the shell is degraded, the iron oxide core start to produce the reactive oxygen species that leads to JNK activation and finally to the apoptosis. We implied that the cytotoxicity should be considered of iron oxide nanoparticles, especially used as a contrast agent, particularly in patients with the liver disease. But maybe we can create a different shape of particles, and that will change their cytotoxicity profile, because while gaining knowledge, it became evident that the surface charge, coating, and the shape are dramatic parameters that are affecting the cellular responses of not only iron oxide particles, but in general, nanoparticles. So we created the cubic ones and cubic cluster small particles. In fact, they were even more toxic. They were already toxic on the cell lines after 48 hours, leading to the profound apoptosis markers, the massive fission of mitochondria retained in lysosomes and led to destabilization of the lysosomal content and leading to the progressive lysosomal leakage and executing apoptosis. Of course, the genetic background mattered as well, that if the cells having the BCL2 elevated levels were present, they were mitigating that to a certain extent, but the particles override still the anti-apoptotic effect and led to the cell death. we went further, even subtoxic doses of the particles that do not induce toxic effects. We take a bigger shell, a particle containing the bigger shell, showed no toxic profile, no oxidative damage, excessive production of the ROS in cells. Those particles still dramatically change the activity of lysosomes, activating the autophagic flux and disturbing the mTOR signaling, indicating that the lysosomes are the master regulator of the responses of iron oxide nanoparticles interactions with cells. Now, a few words about the dosing in MRI imaging. Actually, when you cross-check quite thoroughly the published research concerning how much do you need in order of the iron oxide particles in order to get very nice and sharp contrast, you will find that those nice images you are getting when you are having way higher dose than is recommended therapeutically. What is the current state now? Indeed, all of the iron oxide based nanoparticles as the contrast agents were either discontinued or withdrawn from the market due to various reasons, including the toxicity concerns. We summarized why that happened. We showed firstly that the mechanistic understanding how the particles will be handled by the cells was false from the very beginning. It was thought that because it's iron oxide that it will be embedded into the dietary biomineralization process of the iron. However, although the reality of the uptake of iron oxide nanoparticles has the similar or shared patterns of the uptake pathway, but it's completely different. The lysosomes are working as a central hub that are master regulating the responses of the iron oxide particles, and now it's understanding that nanoparticles at all. Another thing which I didn't stop here much, but we have a lot of research on that, and other groups show similar things, that particles are not naked when they are facing physiological fluids. They are interacting with the proteins, and these proteins are absorbing on the surface and creating so-called protein corona. And not only proteins, there are other biological molecules, but the nickname came from the research on the proteins. And that interaction leads to the oxidative damage of the biological molecules, the surface opsonization, and the immune recognition. The protein corona may screen out the targeting molecules on the particle, and particle loses its ability to go to the targeted delivery as it was intended. Also, this protein corona dramatically changes physical chemical properties, and in case of iron oxide particles, diminish them being as a good contrast agent that leading to changes of the magnetic properties and decaying the contrast. So basically, there are several themes. First of all, mechanisms were overlooked. Then there was overlooked cytotoxicity, and specifically hepatotoxicity of those materials. I didn't stop too much, but they were intended also for the imaging of the tumor lesions. However, it's now becomes evident that the tumor delivery of the particles is failing, and it's not as it was imagined from the very beginning. Poor clinical performance as first meant to be as MRI contrast agents, and all that together led to the clinical failure of those particles. We and other groups show that the ROS-driven cytotoxicity is not a isolated property of iron oxide particles. It's the probably general property of all metallic particles. They all, to a different extent, result in excessive production of reactive oxygen species in cells, depending on the activity of the core. The most active are rare earth oxides. They are extremely active in the production of the reactive oxygen species and in macrophages and Kupffer cells, leading to the pyroptosis, cell death associated with the release of inflammatory markers. And all those particles accumulated in the liver cells, leading via ROS production to various types of cell death, to the oxidative damage, inflammatory responses, and finally there are results showing liver fibrosis. Many times I can see on conferences, especially from the side of physicists and chemists, that, come on, there is a Furahim FDA approved drug, which is based on iron oxide nanoparticles. They must be safe. Indeed, the Furahim is the iron replacement product and is used for the treatment of iron deficiency anemia and has severe anaphylactic reactions that even FDA put it box warning about usage of that drug. That should be carefully considered and in presence of the specialist, and only if another treatment is failing, only to use these intravenously injections of the Furahim. However, still there are voices rising years already that fermoxetol is just a compound name of the drug. That why not to use that as a contrast agent? It's approved. I think it should be carefully considered, especially given in light of the cytotoxicity potential and specifically hepatotoxicity. So, as the Dr. Jekyll and Mr. Hyde from the Stevenson's novel, the nanomedicine has two sides. From one, provides certain benefits, and some formulations, yes, they improve pharmacokinetics. There is one drug that improves overall survival of cancer patients. They have certain reduced cytotoxicity and improved diagnostics. However, drawbacks are not diminished. There is still low delivery to the tumors if we are talking about the tumor delivery. Problem of target is not solved. Hepatotoxicity is poorly investigated. And that all leads to the poor translation of nanotechnology for the real clinical uses. There are even two sides of the research groups. One saying that there is a huge success of the nanomedicine, and other groups are saying that there is a dead end. We think that maybe the truth is in between, because we cannot remove the real success like lipid-based nanoparticles, and with the COVID vaccines, it showed to be very efficient. But we have to realize the challenges of nanomedicines. One initial challenge, and very much criticized by the researchers, is that the focus on only anti-cancer treatment. The majority is for the anti-cancer treatment, and other pathologies are omitted by the development of nanomedicines. And the criteria for design of the nanomedicines becomes evident that they are not very efficient, because there are three major criteria how the researchers are creating some nanomedications for that meant to be for the cancer treatment. It's the, I would say, obsession with these enhanced permeability and retention effects that the particles would be passively accumulated in the tumor. Recent studies show that this is not the case, and probably in humans it's not working. Then enhancing the prolonging the systemic circulation, and in this way the reducing non-specific sequestration by the liver. And reality shows that no, it's not that easy how to say. And the third one is the universal platform that you can use the one formulation for various types of the cancers, for various types of treatments, but indeed the reality shows probably it's impossible to achieve at all. However, we think, and other groups also are resonated with us, that the main problem and challenge is that the development of the nanomedicines in general and nanoparticles specifically is having wrong direction. It's based mostly and coming from physical, chemical, and material properties rather than from the biological perspective. We think that the biological question should be put at the first one. As some example, I want to show you that one. The contrast for the MRI depends on the how good particle has magnetic moment. And iron oxide actually in these regards is not the first thing what you think as a physicist. And if you replace the iron with cobalt, then the magnetic moment is way higher and the contrast is way better. And what people try to do, however, it's known cobalt is rather toxic and very toxic for humans. We had already cases for the previously in history that treatment of cobalt sulfate, cobalt chloride was used for the anemia treatment and was banned due to the non-effective and very toxic. However, it was known already years ago. However, the people started to do those particles and still continue knowing that this is actually dead end. It will never come up to the real application. So to sum up what I told you, there are some knowns about the liver interactions. The iron oxide nanoparticles and the reports are coming more and more showing that there is a liver impairment caused by those particles via oxidative stress, inflammatory responses, the liver dysfunction, etc. However, there are a lot of unknown. The toxicity mechanism remained unclear still, the precise one. The chronic hepatotoxicity studies are not very usual in this area. There are some hepatotoxic effects shown, but they have not been thoroughly investigated in order to be linked to the specific liver disease. And the intrinsic mechanisms are fully elucidated. And specifically the toxic effects in susceptible livers, like from NAFLD, NASH, fibrosis, cirrhosis, are largely omitted in the studies of the iron oxide nanoparticles. I would like to say here that voices are raising that why not to use magnetic nanoparticles for liver fibrosis treatment and diagnosis. So in line of that, the studies on the susceptible livers are wide and very needed. With that, I would like to thank to all of my collaborators, my small group, Cost Action, for giving opportunity to present this our last update on the nanoparticles, and I will be happy to answer your questions.
Thank you, Oleg, for this nice presentation on nanoparticles. Do we have any questions among the audience, or should I start? No questions? Okay. So I can make you one of the first questions that I have nanoparticles is, you showed that in macrophages, for example, these nanoparticles with iron oxide, they trigger apoptosis, but you showed that they increase also propydium iodide, no, which is a marker of necrosis. Are you sure that it's specifically apoptosis, or there are another types of cell death involved? Because then, when you advance in the presentation, you show that some of the main, or let's say there are other ways of cell death, no, like autophagy, always mediated by Jung activation. So, what do you think?
Good point. It very much depends on the composition of the particles and the size. With those small ones, by the way, those two particles, they were drugs previously, but they now withdrawn. The bigger one was reservist, and the smaller ones, this supravist. So we used real drugs that were for MRI, but the supravist was approved in Europe, but not in USA, and reservist was involved. So for those, yes, there is an apoptosis. The smaller ones, they just we have the kinetics of the apoptosis of the annexin 5 accumulation and the propydium iodide entrance. So that one, what I showed, it just was also to illustrate the smaller ones, they already showed profound death, and the bigger one not. But yes, you're right. The others, like let's say cubic ones, they are introducing the lysosomal rupture really fastly, and that is leading to either, depending on the genetic background, either to starting up the autophagic flux to mitigate that, and then shifts to the apoptosis. Yeah, and pyroptosis also involved, but that one is has been shown with the particles that are producing way more higher levels of ROS, like those rare earth metals.
Okay. So thinking about nanomedicine, as you show at the end, there are a lot of I think there is a need of more research, no, and specifically on chronic disease, but also in acute disease. So, for example, if you want to treat a patient that they have this overdose, you would how would you use nanomedicines for acetaminophen, for example, overdose? Would you in which type you would focus?
Honestly, iron oxide particles I would not use at all, because showing the research is coming up that they're quite dangerous to be used. The lipid based, it's another story. That one is way better, and maybe nowadays those, and which we are now actively researching, DNA based nanostructures. Those may have better implications because they are made, then DNA nanostructures, they are made from the biological material DNA itself. Of course, there is a problem. One needs to be careful that they would not introduce immune response, because free DNA, we know it's the immunogenic. But it's a matter of the packaging and creating. But the inorganic ones, and there is also systematic review showing that inorganic particles, they're more toxic to the liver than organic ones.
Okay. Okay. Yeah, this makes sense. I mean, the only thing is that if you change to lipids, probably the hepatocytes will react, no, because they go through the
Yeah.
layer, and also, well, macrophages we will see, but yeah. So it's interesting. I mean, of course, this needs to be studied, no, in detail.
Yes, yes, exactly. I was quite surprised that still with the iron oxide particles, there are a lack of research on the susceptible livers, like with because the idea was, and the thing is that the guys from the chemistry and physics, they try to push those iron oxide particles as MRI contrast agents. By the way, I told that there is no FDA approved drug, but in Europe it's different. EMA doesn't have, but different countries, let's say Reservist still is used in Netherlands.
Mm-hmm.
You can, if you go to MRI facility, they can inject you with that.
Wow. Well, the toxicity is high, no, as you showed.
Yeah. Yeah, and especially I would be afraid of also anaphylactic reactions, as the FDA put it in the box warning for this theory.
Okay. Any other question from the audience?
May I ask a question?
Yeah, Michael, please.
Oleg, very nice talk. You sent up very nice the work with the iron oxide nanoparticles. The liver is the preferential accumulation site for iron oxide nanoparticles or for generally nanoparticles?
For generally nanoparticles, not only iron oxide nanoparticles.
Okay, so can you have any idea what is the reason for that? Why it's accumulating? I mean, the perfusion is very high in the liver, of course, but also the lung has a high perfusion. So why it should accumulate in the liver and not in the lung, for example?
Yeah, so there are some hypotheses on that. One is that the blood flow in the liver, especially in the is lower, and that results into the macrophages there. Kupffer cells are more effectively taken up, so it's slowing down the flow. The blood is slowing down when it goes through the lobules, and that results into the accumulation in the liver cells.
Okay.
That's one thing. Second thing, that specifically to iron oxide dextrane-coated nanoparticles, they are absorbing the proteins from the blood plasma, making the protein corona, and also LDL as well. And LDL, why the scavenger receptor A is involved, because the LDL is the ligand for the scavenger receptor. That's why the macrophages are taking up very efficiently those particles. Yeah, okay. It's like that mostly.
Okay, thank you very much.
Yeah, I think there are also some FDA-approved drugs that they use this siRNA or inhibition of RNA, no, using lipid nanoparticles.
Yes, with lipid nanoparticles, yes, it's different now. They are boosting up.
Because, as you said, they have you can target the cholesterol receptor.
Yeah. But that's also another interesting thing, that lipid nanoparticles, they mostly are from liposomes, liposome containing, and actually some groups are saying that, come on, this is not nanoparticles. It's just the rebranding name of the liposomes.
They're liposomes.
Liposomes were known decades before the nanotechnology started up, but this is gambling with the definitions. As I tried to show at the beginning, because when I did my PhD, the definition of, and this young one is that still. Also definition changed now. The time when I did PhD, the definition of nanoparticle was that you should have unique properties that distinguish that material from bulk and molecules or atoms.
Mm-hmm.
Now it may be improved, and the threshold is changing, like depending on what the people want. So it's also, the definition is not very precise, I would say.
Yes, of course. Any other comment? Another question that I have is what are the advantages, in your opinion, of nanoparticles versus extracellular vesicles? So, for example, to target or delivery of like we just mentioned, RNA or maybe a nanoparticle is better, no? But
yeah.
for extravesicles, they can also deliver.
Thanks for the question. Yeah, should say something good, not only bad about the particles. The scalability and specifically with the DNA nanostructures, you can program very finely the not only the size but shape of the structure. And it's because it's encoded in the DNA, you're basically creating the template, and according to your template, you will generate whatever you want. That's the thing. So the programmability, scalability, and the production of the narrow-sized dispersion of the material.
Okay, I see. Yeah, that's really interesting, because you also have nano vesicles, no? So you have different sizes of vesicles, and as you said, the names are quite the same, no? And it has to be renamed somehow. But it's quite interesting. Any other comments from the audience? Young investigators? No comments? Maribel, you want to say something?
Well, to congratulate Oleg for the excellent work that he is doing right now. I was really struck. Well, he has addressed something that has always surprised me. That is the fact that, for example, chitosan, that we know is hepatotoxic, it was used also as nanoparticle. So I think he has addressed this issue very, very nicely and in a comprehensive way. And I've been very interested about 29 FDA approved nanomedicine and 75 in clinical trials. I do not have experience with these medicines in clinical trials so far here in Europe. At least we have not been involved in any of these studies. But do you think, Oleg, that we should take some special consideration when analyzing or assessing these protocols with regard to safety concerns of kind of follow-up, that we have any special consideration clinical in clinical terms that we should take when assessing these protocols?
No, I do not think that it's really necessary to have some special consideration toward that. Also, there is a notion that people try to kind of bolster this nano and even to invent like nanotoxicity, nano stuff, but it's basically everything goes for the pharmacokinetics and pharmacodynamics. So it's simply the routine pharmacology should be done in a rigorous and reproducible way. That's my suggestion, or my vision. I would say my vision, like that, because we are now trying to wrap up some systematic analysis on the side effects, which I was also very surprised that no one did. On already approved nano drugs, none did at least some certain analysis of the side effects. It cannot be that we have some drugs that have no side effects. Certainly they should have.
Okay. Thank you.
Excellent, Oleg. So I just want to remind everyone that tomorrow it's organized by Malaga, right, Maribel? In Malaga, the 22nd or the 21st hepatology.
The 21st.
21st hepatology session. So tomorrow there is a very nice speakers. So you can, I think the website, well, the link is provided here on the chat. So if you want to go ahead and register, tomorrow there will be a lot of updates in hepatology. And as Maribel said, the chair of ESL is going to be also there. So our next speaker in June will be Jorge Simon. He's from TIC Biogune, and he will present a summary of the data on mitochondrial with mitochondrial damage and acetaminophen, and he will link it to other mechanisms that he has found and he has recently published in several journals, including gut and hepatology. So this will be the 15th of June. So I hope that you can all make it, and yeah, please join us at the 4:30 that date. So that's everything from my side. Do you want to add anything?
No, thank you. Thank you.
Thank you. Thank you, Javier. Thank you, Oleg. Fantastic presentation. Thank you.
Excellent.
Thank you.
Thank you, Oleg.